today, we will learn the chapter 11, cell cycle and cell division. So this chapter is composed of 7 subjects, starting from the all cells derived from other cells. Finally, we will learn the unregulated cell division. Okay, so this is the first subject, the all cells derived from other cells. Okay, so one cells become two cells become four cells become 8 cells like this. So this is the important consequence of cell division. First of all, there is a reproduction. So this is eukaryotic yeast cells dividing by budding, or there is another sexual reproduction comes from the sperm and egg. After they fertilized, they will become the babies like this. And also another consequence is growth. For example, this is the root tissue, 뿌리 tissue. So cell division contributes to growth of this root tissue. One cell become two cells become four cells become 8 is like this. Another consequence is the repair and the regeneration. For example, this is the lizard tail. So after the tail was cut, so there will be the tail will be regenerated is like this. Regenerated is like this. So this is also important consequence of cell division. And also prokaryote, especially the prokaryote, they produced daughter cell by binary fission. In Korean, we call it as 이분법. Okay, so one cell became two identical. They are same. Two identical daughter cells. Okay, one cell become the two identical daughter cell. Okay, so during the binary fission, so DNA replication start from the origin of replication. After that, they will make the replication fork. After finally the DNA is segregated is like this. They make the two identical DNAs. Finally, also there is happen cytokinesis. Okay, so one cell divided by fission, cytokinesis. So after that, it will make the two cell is like this. Okay, so this is the photograph to show the prokaryotic cell division. Okay, here you see the one cells become the two cells is like this. So this is we call it as cytokinesis. Okay. How about the eukaryote? Eukaryote as a cell cycle, starting from the G1, S, G2, M phase. Okay, so G1, S, G2 phase. Especially we call it as G1 plus S and G2 phase. We call it as interphase. And also there is an M phase is like this. So G1, S, G2, we call it as interphase like this. Okay. Okay, so during the G1 phase, okay, cellular contents excluding chromosome are duplicated. G1 phase is there are duplication is happens. One protein will become two protein, one carbohydrate become two carbohydrate, one lipid become two lipid. But chromosome usually replicated during the S phase. At that G2 phase, cells double-checked the duplicated chromosome for error. So cell checked whether there is error in the chromosome or not. So if they find some error, they stopped here. The cell cycle does not progress to the M phase. They are stopped here. Okay? Okay? There is M phase. We call M phase. So M phase is composed of those two important steps. First one is mitosis, and second one is the very small phase. We call it as cytokinases. So mitosis followed by cytokinases. So nuclear division happens during the mitosis. After that, cytokinases means that cell becomes two cell is like this. Okay, so M phase is composed of mitosis and cytokinases. Okay, and also specific signals trigger the transition from one phase to another. The first one is cycling proteins. So, for example, cycling proteins made only at certain point during cell cycle. Especially the G1 phase, cyclin D is synthesized. Okay, how about the S phase? Cyclin E and also some of the cyclin A. Especially in also G2 phase, cyclin A is synthesized. Finally, during the mitosis, cyclin B is synthesized and disappears, is like this. Okay. Second one is the cyclin-dependent kinase. We call it as CDK. CDK is controlled cell cycle progression. Okay, especially this is one example. So this is a cyclin. Some specific cyclin is made at specific phase. Okay, at that time, but CDK is always present, but its active site is not exposed. So after their binding, cyclin binding changes CDKs, exposing the active site is like this. After that, several protein substrate can bind to the enzyme site, active site. After that, this protein substrate is phosphorylated is like this. So this actually this phosphorylated proteins regulate cell cycle is like this. Okay, so cyclin, CDK. Okay, so they after their bindings, so they expose the active site. After that, substrate is bind to here. After that, ATP is used. So ATP is changed to ADP. After that, this protein substrate is phosphorylated. So actually, this protein is regulate the cell cycle. It's like this. Okay, and also there are several cyclin CDK complexes. For example, especially the G1 phase. There is a cyclin D1 CDK6 complex or cyclin D1 CDK4 complex or cyclin D2 CDK6 or cyclin D2 CDK4 is like this. How about G2 phase? G2 phase, there is a cyclin A and CDK1 complex. How about M phase? There is a CDK1 and cyclin B complex. there are many different cyclin CDK complexes acting at different stage of cell cycle like this. Okay? Okay. Okay. So in this slide, we will learn the third subject, eukaryotic cells divide by mitosis. Okay, so we learned mitosis. Previously, we learned mitosis. Okay, so mitosis is so this is mitosis. Okay, so first of all, we will learn the chromosome and sister chromatid. Okay, so during the interphase, starting from the G1, S, G2 phase, okay, during the interphase, chromosome looks like the thread-like. In Korean, we call it as 실타래. Thread-like structure is like this. And dispersed throughout nucleus is like this. But during the M phase, during the M phase is composed of the mitosis followed by cytokinases. During the M phase, DNA and protein in each chromosome forms highly compact structure is like this. Okay, highly compact structure is like this. Okay, so this is the chromatid. So after the chromatid is replicated, so it will make the sister chromatid. Okay, so and we also call it this one as chromosome. Is like this. Okay, totally we call it as this is chromosome. It's like this. Okay. So usually the DNA is packed into a very compact chromosome. So this is the DNA, and DNA is make the nucleosome, and nucleosome make the chromatin, and chromatin will make the compact chromosome is like this. So during my epigenetics course, maybe Maria and Marin already took my epigenetics course. At that time, I teach the euchromatin and heterochromatin, but today I will not cover. Anyways, DNA is packed into a compact chromosome is like this. Okay, let me see some details. So DNA wraps around the histone. This is histone proteins. Okay, but you don't have to memorize the specific histone name. Okay, so DNA wraps around the histone to form nucleosome. We call it a nucleosome, and nucleosome is like this. So DNA will make the nucleosome is like this. After that, this nucleosome folds up to produce 30 nanometer fiber. So this is a diameter. So it will make the 30 nanometer the fiber. We call it as a chromatin. So chromatin will become scaffold associated chromatin, and scaffold associated chromatin will become condensed chromatin. And also during mitosis, especially during mitosis, chromosome will become very compact. Structure is like this. They have the 2 arms, 2 legs is like this. Very compact chromosome is like this. Okay. Okay. Now I will teach a very important terminology, mitosis. Subject 3 is the eukaryotic cell divide by mitosis. So in this slide, I will teach is mitosis. And also, here, the fifth subject mentioned about meiosis. At that time, I will teach the meiosis, okay? Let me start from the mitosis. So mitosis is after interphase, after G2 phase, they go to the mitosis. Okay, so during the mitosis is subdivided into the prophase, prometaphase, metaphase, anaphase, and telophase. Okay, so during this step, one nucleus will become the two nucleus, but they are genetically identical to each other. Okay, so mitosis is produced two nuclei that are genetically identical to each other. They are subdivided into prophase, prometaphase, metaphase, anaphase. Finally, there is a telophase is like this. Okay. Let me see some detailed information. How about the mitosis? Prophase. So this is the interphase. So this is nucleus. And this very big dot, we call it as nucleolus. Okay. Okay. But we cannot see the chromosome structure here. But during the prophase, chromatin condense into chromosome. They will make some structure is like this. Okay. So this is the DNA. So this DNA is condensed. So they will make the chromosome is like this. So previously we learned that this DNA is condensed into the compact chromosome during mitosis, especially prophase. So at that time, so there is a two centrosome. Centrosome, we call it as a 중심체. Centrosome, we call it as centrosome. They are composed of the centriole plus microtubule. This is microtubule. We call this centriole plus microtubule. We call it as a centrosome. Okay, this centrosome, they are located in the center, but they move to the opposite poles. Is like this. Okay. After that, there is the prometaphase. So prometaphase, in Korean we call it as a 전중기. Prometaphase. So during prometaphase, nuclear envelope is breakdown. Okay, so after that, centrosome, they are composed of the microtubule. Okay, especially this microtubule, we call it as the kinetochore microtubule. So, for example, this is the chromosome. At the center, there is the centromere region is located at the center. Next to the centromere, there is a kinetochore. So usually the microtubule, especially the kinetochore microtubule, is bind to the kinetochore, not centromere. Okay. So usually the kinetochore microtubule, okay, appears and attach to the kinetochore. So this is a pro-metaphase. So you can see it here. There are lots of the green structure. This green is microtubule. So they are attached to the chromosome. Is like this. So previously, but in the prophase, so this is inside the nucleus. There is no microtubule. There is no kinetochore microtubule, but pro-metaphase, you can see there are lots of kinetochore microtubule. They appears and attach it to kinetochore. Okay. Next page, we call it as metaphase. Okay. During the metaphase, chromosome is lined up. So this plate, we call it as metaphase plate, okay, around at the center. So at that time, chromosome lines up. Lines up means that one chromosome, nearly second chromosome, like this. This is, we call it as lines up. Okay? Lines up. So chromosome lines up around the metaphase plate. After that, there is we call it as anaphase. Okay. So chromosome is composed of a sister chromatid. Okay, so they are paired sister chromatid, but during the anaphase, paired sister chromatid are separate. So this is a chromosome. Chromosome consisting of one chromatid. After this chromosome is replicated or duplicated, they will make the sister chromatid, and sister chromatid is like this. After that, during the anaphase, these sister chromatids are separated. After that, they will make, we call it as a daughter chromosome. Okay? So during the anaphase, paired sister chromatids separate. After that, daughter chromatids begin to move toward each pore. It's like this. So this is the anaphase. Okay, you can see lots of kinetochore microtubule. Okay. So after that, finally, there is the telophase. So telophase is the each daughter chromosome or daughter chromosome reached at each pore. After that, nuclear envelope reforms. After that, chromosomes start to decondense. You can see that decondense. Okay, their structure is the decondensed like this. Okay, so this is the telophase. So finally, after mitosis, cytokinesis happens. Okay. For example, here we learn that again. M phase is composed of mitosis After that, followed by cytokinesis. Cytokinesis is cell division happens. Okay, if you see here, so for example, animal cell, we can divide it into animal cell and plant cell. So this is the cytokinesis of animal cell. So usually the contracted ring forms after that pinches. They will make the one cells and pinches two cells is like this. So this is happen in the animal cell. How about the plant cell? So plant cell, so one cell. During that time, so this vesicle comes from the Golgi apparatus. So this vesicle comes from Golgi apparatus. After that, these vesicles fuse each other. So these vesicles fuse each other. Okay, so after that, they will make the cell plate is like this. So they will make one individual daughter cell is like this. So this is the cytokinesis. So after that, we will learn the cell division. First subject, cell division plays important role in sexual life cycle. Okay, so previously we learned that cell division is very important role in cell reproduction. So we will see some specific example starting from the asexual reproduction. So previously we learned that some clone. So this is one clone, the other clone, one clone. They are identical, same. Okay, this is, we call it as clone. Okay, so first of all, we will learn the asexual reproduction. At that next slide, we will learn the sexual reproduction. So asexual reproduction is based on mitosis. Okay, figure 11-13 to show that asexual reproduction happens on large scale. So in this forest, some tree, the name is aspen tree, all of tree is arose from single tree. It means that they are all of the tree is clone each other. For example, if you see the details, so aspen trees have a shoot, they share each other. So in the bottom, we cannot see it, but in the bottom, they share the root each other. So they will make another clone. They will make another clone, another clone, another clone, another clone, another clone is like this. Okay. So all three in a stand may be clones of a single parent. So this is, we call it as asexual reproduction. Sexual reproduction means that, for example, there are several sexual reproduction, especially this is some bird. Okay, for example, male has sperm, our female has egg. Okay, this sperm and egg, we call it as gamete. Gamete in Korean, we call it as 배우자. Sperm and egg, we call it as the gamete. Okay, each gamete are created by meiosis. We will see some details, meiosis, okay? Meiosis and meiosis. So each gamete, for example, sperm and egg, contains only one set of chromosome. So we have the duplicate chromosome, okay? But they contains only one set of the chromosome, haploid. Our human we have the 2n is 46 is like this, but our sperm and our egg, they only have N23. N23 is like this, okay? They only have one set of chromosome. We call it as haploid. So each haploid fuse to form a zygote. They will become the 2N. We call it as zygote. In Korean, we call it as 접합자. Okay, this is the 2N status. It's like this. So this is, we call it as a sexual reproduction. Different from the asexual reproduction. Asexual reproduction is based on mitosis. However, sexual reproduction is based on the meiosis. Okay? That's why we will see in 5th chapter, we will see the meiosis. Okay? Meiosis. In Korean, we call it as 감수분열. Okay? During the interphase, there was one nucleus was located here. But however, during meiosis, one nucleus will become the two nucleus. Okay? One nuclear division happens. After that, meiosis 2 procedure, one nucleus will divide into two nucleuses like this. It means that they are composed of the two nuclear division. But DNA is only replicated only once. When DNA is replicated, during the interphase, DNA is replicated. Okay? 김한 수업 잘 듣고 있어? 직내 알겠지? 오케이. 오케이. 김민지 수업 잘 듣고 있어? Okay. How's the Marin and Rhea? How's the lecture? It's good? Okay. How many classes do you take this semester? 5? Okay. 6? Oh my God. Okay. So now is maybe next week we have the exam. Not next week. Okay. The following. Okay. 지금 선생님 수업 잘 듣고 있어? 자고 있는 것 같은데? Okay. Wake up. 수업 잘 듣고 있어요? Okay. Okay. I found this. Some of students fall asleep, so just I have a break like this. Okay. 그러니까 제가 이제 결혼할 때 저 와이프가 중학교 교사거든요. 중학교 교사인데 결혼을 좀 엄청 오래전에 했는데 어렸을 때 결혼해가지고 근데 이제 와이프가 그때 교사였어요. 그래가지고 그때는 이제 2004년도에 결혼했는데 그때는 중학교가 토요일에도 수업을 했어요. 이제 와이프가 이제 토요일 날 자기 반 담임 애들이 다음 주에 우리 결혼식 때 노래 불러줘야 되니까 아이스크림을 사갖고 오라고 했어요. 이제 저는 1시쯤에 가가지고 아이스크림 사주고 이제 저는 나눠주고 바로 그냥 가고 와이프는 이제 애들 이제 종례하고 했는데 와이프가 내가 가자마자 애들 바로 찾아와가지고 그 선생님은 외모로 사람을 판단하지 않으시는군요. 그런 일화가 있었던 거. 그래서 듣고 보니까 그 아이들이 와이프가 완전 그 안목을 무시하는 것 같다고 다 빵점 주라고 애들 다 이제 그런 일이 있습니다. 오케이, meiosis. So there are two nuclear division, but DNA is replicated only once during the interphase. They only have one interphase here. Okay, so but reduced chromosome number from the diploid to haploid. From diploid to haploid is like this. Okay. So meiosis is composed of prophase 1. So we will see several procedures starting from chapter slide 17, 18, 19, 20, 21, and 21 is like this, okay? Okay, let's start from the 17. Okay, 17, okay, meiosis 1. The starting point is prophase 1. Okay, so this is the early prophase 1, mid prophase 1, and late prophase 1. Okay, chromosome begin to condense. So after following the interphase, the chromosome begin to see or begin to condense. Okay, so finally they will make the synapses. Okay, so chromosome, they are homologous. This is homologous chromosome. Okay, because we have the 2N. Okay, for example, if this is chromosome 1 has two chromosomes. Homologous chromosome is like this. Okay, in each chromosome, this is the sister chromatid, and this the sister chromatid. But for example, this sister chromatid, cis chromatid. We also see this chromatid as the non-sister chromatid. This is also comes from the homologous chromosome, but they comes from the different chromosome. They comes from the homologous chromosome. So this is sister chromatid, and this is recorded as non-sister chromatid. Lengthwise pairing of homologous chromosome recorded as a synapsis. So they are doing the synapsis is like this. Synapsis is like this. Or the chiasmata, so region of attachment that forms between non-sister chromatid. Okay, for example, this red chromosome maybe comes from the mother, and this blue chromosome comes from the father. Okay, so this each chromosome we call it as non-sister chromatid. Okay, and also the chiasmata or chiasma. Anyone is okay. Chiasma or chiasmata. This is the region of attachment that forms between the non-sister chromatid. Okay, so this is chiasma or chiasmata. After that, usually prophase 1 crossing over is happen. So after that, they will make the recombinant chromatid is like this. Crossing over happens, so they will make the recombinant chromatid is like this. exchange of genetic material between the non-sister chromatid at chiasmata. Okay, so this is happened during the prophase 1. How about pro-metaphase 1? So this is pro-metaphase 1, late prophase 1 and the pro-metaphase 1. So especially the pro-metaphase 1 nuclear envelope is breakdowns like this. So breakdown of nuclear envelope. After that, they go to the metaphase 1. So during metaphase 1, okay, random alignment of homologous pairs of chromosome along metaphase plate 1. For example, red chromosome comes from mother, and the blue chromosome comes from father. Okay, so red chromosome is located at the left side. In this case, this blue chromosome is located in the right side. How about another chromosome? In this chromosome, blue comes from the father, is located in the left side, and red comes from mother, is located in the right side. In this case, we call it as a random alignment of homologous pairs of chromosome along the metaphase plate 1. Okay, so previously in mitosis, we learned that metaphase, okay, at that time I say that chromosome is nearly lines up, is like this. However, in metaphase 1, they are pairs each other. They are random. They are each homologous chromosome is located along the metaphase 1 is like this. Next to next, next to next, next to next is like this. This is not lined up, lines up. This is next to next, next is like this. Okay. After that, there is the anaphase. Okay. During the anaphase 1, each homologous chromosomes move to the opposite pole. How about mitosis? During mitosis, paired cis chromatid is separate. After that, they will make daughter chromosome. So daughter chromosome begin to move toward each pole. However, during meiosis, there is no separation. There is no separation, but each homologous chromosome moved to the opposite pole. After that, telophase 1, nuclear membrane envelopes reforms. This is telophase 1. Finally, followed by cytokinases. So cytoplasm of original cell split into two daughter cells like this. This is, we call it as cytokinases. Okay. After that, they will go to the prophase 2. Okay. So now we finished the meiosis 1, telophase 1. After that, there is cytokinases. Okay, so after that, we will start the meiosis 2. Okay. Okay. So this is the meiosis 2, especially the prophase 2. So chromosome condense again. Next that, there is the metaphase 2. So during the metaphase 2, chromosome is lines up, like the mitosis. They are lines up. Okay. During mitosis, during metaphase, chromosome is lines up, is like this. However, meiosis 1, metaphase 1, they are the homologous chromosome, they are not lines up, they are next to each other. Okay. However, in metaphase 2, they are lines up along the metaphase plate 2 is like this. After that, they will go to the anaphase. So anaphase 2 is important thing is each chromosome is separate, like the mitosis. Move to the opposite pole. Move to the opposite pole. At that telophase 2, nuclear envelope reforms. Nuclear envelope reforms. After that, followed by cytokinases. Cytoplasm or original cell split into 2 daughter cells is like this. So this is we finish the meiosis. Okay. So meiosis is composed of the meiosis 1 and meiosis 2. 2 nuclear division happen. 1 nuclear become 2. Another, and also each nuclear divided into 2. 2 nuclear division, but only one time DNA is replicated. Okay, so initially they start from the 2N, but finally they will make the N. Okay, like this. Okay, so they are composed of the meiosis 1 is composed of prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1 is like this. And it's meiosis 2 starting from the prophase 2, metaphase 2, anaphase, and telophase. After cytokinases happen is like this. Okay. And also we will learn some important step during the meiosis. Okay. During the meiosis, usually lots of genetic variation is happens. Okay. For example, Doyeon, how many brothers do you have? One sister. Okay. Youngwon, how many brothers or how many sisters do you have? One sister. Jayan, how many sisters do you have? Huh? 없어? 오케이. 박주원, 몇 명 있지? One younger brother. Okay. How about 지혜? Older brother. How about 종곤? 남생 한 명? Okay. If you have the, for example, Doyeon have the brother, they receive the chromosome from mother and father. They receive the same chromosome from mother and father. Your brother, if you have brother, your brother also receives same chromosome from father and mother. But their looks different because genetic variation happens. Even though they have same father and mother, usually your sister is look different, isn't it? Because of genetic variation. Okay. First genetic variation is crossing over. During meiosis, usually the crossing over happen, especially the prophase 1. So they will make the synapses. At that they will make the chiasma or chiasmata. This is a lesion. This is non-sister chromatid. after that, crossing over happens. So usually crossing over forms genetically different, diverse chromosome is like this. This is a recombinant chromatid is like this. Okay? That's why your brother or your sister is not identically like you. Okay? And another thing is the random alignment of homologous pair. Okay, during the meiosis, we already covered it here. So usually the metaphase 1, random alignment of homologous pairs of chromosomes happens like this. Okay, for example, here, this is the one possibility 1, and this is possibility 2. For example, this blue comes from father, and maybe this red comes from mother. But this is possibility 1, but they will make combination 1 or combination 2 is like this. However, random alignment happen. So in this case, they will make this combination, or they will make this combination is like this. Okay, so this random alignment of homologous pairs of chromosome happens. So they will do independent assessment that they will make the genetic variation is like this. So we will see this concept in the chapter 13 next Monday. We will see some detailed information of this one. Okay. Today I will not cover, but during the meiosis, especially the metaphase 2, okay, in the midterm exam, you have to know, especially the metaphase 2, random alignment of homologous pairs of chromosomes is happens. Which phase? Metaphase 2, okay? How about crossing over? When does it happen? Kim Do-yeon? When does it happen? Jo Young-un? When does it happen? Which phase does it happen? I already mentioned that during meiosis, especially the prophase 1, crossing overs happen, okay? Okay, now we will cover non-disjunction. So non-disjunction happens during meiosis 1, okay? For example, during the meiosis 1, especially metaphase 1, homologous chromosomes, they are not lines up, they are next to each other. So during the anaphase and telophase, homologous pairs may fail to separate at anaphase 1. So both chromosomes go to the only one cell is like this. Okay. So usually each cell have the one chromosome is like this. But in this case, they did not receive chromosome, but in this case, they received one more chromosome is like this. So it will make aneuploidy. They will have the extra chromosome is like this. So finally, this sperm, they are lacking one chromosome. This sperm, they have extra copy of chromosome. So after fertilization happens, so they will make the trisomy zygote, or in this case, they will make the monosomic zygote. Okay, the trisomic zygote happens very frequently, especially the trisomy 21. For example, chromosome 21, only the non-disjunction happens. So especially chromosome 21 is go to the only one cell. So finally, they have chromosome 21, but they have 3 pairs. It's like this. So this is the Down syndrome. They have 21 chromosome, but they have 1, 2, 3 is like this. Non-disjunction happens. Okay, so this is the non-disjunction usually happen. Sometimes happen if their mother's age is higher. At that times, non-disjunction happens. If the mother's age is higher, there are lots of the possibility their baby have the Down syndrome like this because of non-disjunction happen during meiosis. Okay. Now we will cover the fixed subject. Cell death is important in the living organism. So usually the cell is damaged or starved. Cell is damaged or starved of oxygen or nutrient. Usually cell wall is burst, swell and burst is like this. So usually the cell contents are released to extracellular environment and they will cause inflammation is like this. And this tissue is specially, we call it as they did necrosis. Necrosis in Korean we call it as 개사. Okay, this is one example of cell death. However, there is another programmed cell death is there. Especially we call it as apoptosis. This is programmed cell death. So if cells are no longer needed, for example, connective tissue between finger. Okay, previously when we were baby, when at 41 days, our finger is attached to each other. But at that time, so the between here, apoptosis is happen. So it will make the finger is like this. Okay, day 56. So apoptosis happens. So they will make the finger is like this. So if cells are no longer needed, especially the connective tissue between fingers of fetus, they commit suicide by activating cellular intercellular death program. Okay, today, but we will not cover in details. But important thing is that apoptosis programmed cell death. So if cells are no longer needed, okay, so they will commit suicide. So after that we have fingers like this. Okay, but there are several death signal is there, but today we will not cover in details. Okay. Okay, so today we finish